Hello, my name is Sophia Forbes, and I will be presenting my presentation on the diphtheria toxin. So, diphtheria is caused by Corynebacterium diphtheriae, which is a club-shaped gram-positive bacillus. It can either cause respiratory or cutaneous infection, but respiratory diphtheria is the classic and more dangerous form. A key point is that not all strains produce the diphtheria toxin. The toxin production depends on whether the bacterium has been infected by a beta Corynebacteriophage that carries the tox gene. When this phage integrates into the bacterial genome, it can convert a non-toxin producing strain into a toxin producing strain. This process is called lysogenic conversion. In respiratory diphtheria, the bacteria usually stay localized in the throat, but the toxin causes local epithelial damage. This contributes to the formation of the thick gray pseudomembrane. The membrane can make swallowing and breathing difficult, and in severe cases, it can obstruct the airway. If the toxin spreads systemically, it can also damage the heart and peripheral nerves, leading to myocarditis and neuropathy. Diphtheria toxin is a phage-encoded AB exotoxin. So it's produced as a single-chain polypeptide of about 535 amino acids, having a molecular weight around 58 to 60 kilodaltons. And so even though the toxin begins as a polypeptide, it has different functional regions. Fragment A contains the catalytic domain. Fragment A contains the catalytic domain, which is the enzymatic part of the toxin. Fragment B contains the translocation and receptor binding regions, which help to deliver the toxin attached to host cells and bring the catalytic fragment into the cytosol. The three major domains are the C, T, and R domains. The C domain is the catalytic domain and performs ADP ribosylation. The T domain responds to the acidic endosomal environment and helps move this catalytic fragment across the endosomal membrane. The R domain binds the host cell receptor, which I'll get into further in further slides. The toxin also has to be proteolytically nicked into fragment A and fragment B at this bridge here. And these fragments remain connected by a disulfide bond until reduction allows fragment A to separate. This enables translocation. Once fragment A reaches the cytosol, it uses NAD+, shown here, as the ADP ribose donor to modify EF2. Now, what does that mean? As you can see in this image here, this is NAD+ nuzzled in this active site in the catalytic domain. So, the specific biochemical pathway that's affected by diphtheria toxin is eukaryotic protein synthesis. More specifically, the toxin blocks the elongation stage of translation. So, during normal translation, elongation factor 2, also called EEF2 or EF2, helps move the ribosome along the mRNA. This movement, being called ribosomal translocation, allows the ribosome to shift to the next codon so that the growing polypeptide chain can continue to elongate. EF2 normally functions as a GTP-dependent elongation factor. So this means it uses energy from GTP hydrolysis to help drive ribosome movement. When diphtheria toxin modifies EF2, this translocation step can no longer occur efficiently. As a result, translation elongation stops, and the cell cannot continue producing proteins. Now, what's the actual mechanism here? Well, it begins when the diphtheria toxin binds to the HB-EGF precursor receptor at the host cell surface, seen in step 1. After binding, the toxin enters the cell by receptor-mediated endocytosis, or this clathrin-coated pit. Inside the endosome, the pH drops, and proteases partially cleave the bond between the different subunits, and these acidic conditions trigger a conformational change in the toxin. The translocation domain then helps move fragment A across the endosomal membrane into the cytosol. Once it's in the cytosol, fragment A acts as the NAD+ dependent ADP ribosal transferase. It takes the ADP ribose group from NAD+ and transfers it onto EF2. The specific target site on EF2 is diphtheriae, which is a modified histidine residue. This modification inactivates EF2. It causes a conformational change that reduces its ability to cause translocation. Since EF2 is required for ribosomal translocation, during elongation, the ribosome can no longer move properly along the mRNA. The result is a shutdown of protein synthesis. So the disease symptoms are directly connected to this biological mechanism. Once EF2 is inactivated, affected cells cannot make proteins efficiently. Proteins are needed constantly for membrane maintenance, enzyme function, metabolism, repair, and survival. Without new protein synthesis, cells become injured and eventually die. In respiratory diphtheria, this damage occurs mainly in the throat and upper airway. The toxin injures epithelial cells, causing inflammation and tissue necrosis. Dead epithelial cells, bacteria, fibrin, white blood cells, and different inflammatory debris collect on the surface and form this thick gray pseudomembrane. This pseudomembrane can adhere tightly to the throat tissue and physically narrow the airway. This is why respiratory diphtheria can become life-threatening even when the bacteria remain localized. Now, if the toxin enters the bloodstream, it can affect distant tissues. The heart and peripheral nerves are especially important targets. In the heart, toxin-mediated injury can cause myocarditis, abnormal rhythms, or even heart failure. In peripheral nerves, it can cause neuropathy, weakness, difficulty swallowing, or paralysis. So, the overall disease pattern follows one main chain. Diphtheria toxin inactivates EF2, protein synthesis stops, cells die, and that leads to local airway damage or even systemic complications. Thank you for your time, and here are my references.